Hello dear viewers, myself Nasir and welcome to Mr.Zoologist YouTube channel. Today we are going to discuss about the phosphorylations in detail. Phosphorylation of CDK molecules. CDK, you might be familiar with it. These are the proteins. These are cyclin-dependent kinases. CDKs must be phosphorylated at specific amino acid residues in order to be activated. At the same time, inhibitory phosphorylations of other amino acid residues must be removed. These inhibitory phosphorylations are removed. They are necessary to be removed these phosphates from the specific CDKs because the regulation of the CDK is interdependent between the hypo and hyperphosphorylation. The stimulatory phosphorylations are made by a serine and threonine kinases termed as CAK, stands for CDK activating kinase. CAK itself is a cyclin-dependent CDK complex which phosphorylates the activation loops, often called T loops of CDKs, which in turn phosphorylates the downstream targets. It means that CDK activated cyclin complexes will phosphorylate the further molecules which are essential for progression of the cell cycle at different levels of the stages. So cyclin CDK complex is in your view, that is the top structure that is showing the p state alpha helix, and on the downstream is the activation loop that is necessary for the activation or binding of the CDK with E-type cyclins. So if we talk about the structure of cyclin CDK complexes, the complex is formed by CDK2 with its partner cyclin. In each case, the cyclin activates the CDK molecule through CDK catalytic site and directs the resulting activated complex to assign substrates for phosphorylation of the upcoming or downstream targets. During the late G1 phase of the cell cycle, cyclin E directs CDK2 to substrate proteins or phosphorylate proteins that must be phosphorylated in preparation for entrance into the S phase. Another segment, orange, is involved with the cyclin E's association with centrosomes. The p-state alpha helix is present in all CDKs and is essential for binding of cyclins. The activation loop, sometimes termed as T loop, must be phosphorylated on threonine residue by a CDK activating kinase CAK in order to function its catalytic activity or to enable the CDK activated. The effects of inhibitory phosphorylations affecting CDK2 and CDC2, which are removed by a class of phosphatases called CDC25A, B, and C, they play a critical role in triggering the G2 to M transition. So, if we talk about the phosphorylation of retinoblastoma protein, is a tumor suppressor which plays a central role in the negative control of cell cycle and in tumor progression. It has been shown that RB retinoblastoma protein is responsible for a major G1 checkpoint, blocking S phase entry and cell growth. It controls the cell by phosphorylation of its amino acid residues. PRB is essentially unphosphorylated when cells are on GO or resting state, becomes weakly phosphorylated, hypophosphorylated on a small number of serine and threonine residues after entrance into G1 phase. It becomes extensively phosphorylated or hyperphosphorylated on a much larger number of serine and threonine residues as the cell passes through the R point into the S phase. Once cell has passed through the R point, PRB usually remains hyperphosphorylated, means cell extensive phosphorylations kept
Once
on the
cells have
retinoblastoma
passed
protein
through the
throughout
R
the remainder
point,
of the cell cycle. After the cell exits
PRB
mitosis, the
usually
phosphate
remains
groups on PRB are removed by the enzyme termed
hyperphosphorylated.
protein phosphatase type
Through
1. So these are the
in this
cyclins and
diagram,
CDKs levels at the
it
different
is
stages of the cell cycle.
shown
As you can see
that
the diagram, the D, CD,
PRB
CDK4 and 6 are
phosphorylate
abundant in the
weakly
G1, and the
during
CDK2 is replaced by CDK4
G1
6 and at the R
phase.
point, and
But
after
once
that
the
CDK2
cell
binding
passes
with the
through
A-type
the R
cyclins
point,
replacing E-type
the
cyclins, and after that
phosphorylation
A-type
of
cyclins
PRB
kept on with the
becomes
replacing CDK2 with CDC2 or
hyper
CDK1.
level.
CDC2 is also known as CDK1, cyclin-dependent kinase 1. So, the phosphorylation state of the PRB red circle is closely coordinated with cell cycle advance. As cell passes through the M to G1 transition, where virtually all the existing phosphate groups are removed from the protein retinoblastoma, leaving it in an unphosphorylated configuration. As cell progresses through the G1, a single phosphate group is attached at any one of 14 different phosphorylation sites of cyclin D CDK4/6 complexes, yielding hypophosphorylated PRB. However, when cells pass through the restriction point, cyclin E CDK2 complexes phosphorylate PRB on at least 12 more sites, replacing it in hyperphosphorylated state throughout the remainder of the cell cycle. The extent of PRB phosphorylation remains constant until cells enter into M phase. So that was the video for today. We will meet in the next video. Till then, goodbye.